Hello everyone, and welcome to this audio podcast. In this series, audio podcast series, the Storm Focus V3. This audio podcast will be in and around T cell activation, allo recognition, and allo response in aligned to organ transplantation. This is the Shientist with another Storm Robotics production surrounding observing chemokine and chemokine receptor expression in acute renal allograft rejection, or AAR. And this is another Storm Robotics VenDip. And so in following the initial audio podcast in this series, to follow on from that, or follow on from there, let's begin with the definition. Let's begin with definition of self in context of T cell activation, allo recognition, and allo response, and organ transplantation, and in around renal transplantation. So for T cells in context of self, it was works by Waldmann in Waldmann in the sorry Waldmann in and around in around in the mid 2000s, around 2006. In 2006, had highlighted or suggested some context in works surrounding co-receptor and co-stimulation blockade of mixed chimerism and tolerance without myelosuppressive conditioning, in those and surrounding the self, and how self consists only of major histocompatibility complex, or peptide complexes. Um, and further, it has been highlighted that self consists only of peptides found in the thymus, and that all other organs or tissues are ignored. For B cells, works by Matzinger had highlighted that self and cell surface and soluble molecules are also in place in context of housekeeping, proposing that housekeeping antibodies to intracellular components might might help clear cellular debris. Ultimately, the entire model, sort of the entire immunological bases boils down to the idea that the immune system regards a certain subset as self, and a particular fraction of the rest of the rest of the universe in context of physiology, and at least in context of physiology and immunology, or immunobiology, is linked, is in this context is an external. An external is and would be foreign. The immune system thus classifies antigens into four particular categories. So number one is visible self, where bodily structures to which the immune system is tolerant. Number two, invisible self, where bodily structures to which it is not tolerant, but to which it does not normally respond. Visible non-self, structures to which the immune system normally responds, and invisible non-self, which is where there's non-immunogenic or immunological structures or bases to which it does not respond. Works again by Matzinger in Matzinger's works in the mid 90s, or mid 90s, in a 1994 publication around tolerance, danger, and the extended family, in the journal, where works was published in the annual review of immunology in 1994 by Matzinger, and so the problem with or the disadvantage with the sort of classification as identified by those four sort of categories of in context of the immune system or immunology and the antigens. The problem or the disadvantage is that this kind of classification is that it is virtually impossible to uncover the rules by which the immune system could make necessary could make necessary distinctions, or can make necessary distinctions. And so, for example, in works by Waldmann again, the idea that the cell for T cells consists of common peptide or major histocompatibility complexes, or MHC complexes, begs the question: what happens when and during the course of normal life, and the peptide and the during the course of a normal life, where peptides change. There's works by in works by Matzinger again. There's some basis in and around what's been referred to in context again of immunology or the immune system, in context of the immune system and in and around immunology, what has been referred as ignorant or T cells, ignorant T cells, and that should often, where ignorant T cells should often be activated by cross-reactive environmental antigens, and autoimmunity should be frequent and irrevocable event, rather than the rare occurrence. So in self or non-self models, T cells must be almost trained, if you like, to distinguish the difference between the foreign and self antigens. And in an immune system that's poised for danger, the T cells need only to identify antigen presenting cells from everything else. So in a nutshell, if any T cell specific antigen presenting cell, antigen presenting cell antigen were added, and if APCs, antigen presenting cells, were the only cells able to activate T cells, and then the only T cells that would be stimulated during an immune response would be those specific for new antigens presented by APCs or antigen presenting cells, and again works by Matzinger in 94 highlight some further aspects in and around this. Initial recognition of processed antigen by T cells is through the T cell antigen receptor. Accessory molecules further link the antigen presenting cell APC and T cell, leading to a stronger cell interaction. For example, CD4 binds to the constant region domain of class 2 MHC molecules, while CD8 binds to class 1 MHC molecules. There's works by Lydiard and team in 2001 who look at works in some depth around MHC, the major histocompatibility complex classes 1 and 2. CD4 positive T cells can differentiate down at least two pathways to become inflammatory T cells or Th type 1 cells, or Th2 type Th type 2 cells, which support or aid B cells. T helper cells type 1 are predominantly involved in mediating inflammatory immune responses through the activation of macrophages, while Th2 cells are predominantly involved in the introduction of humoral immunity through the activation of B cells. Th type 1 cells produce cytokines that primarily act on macrophages. So, for example, in and around interferon gamma and tubular necrosis factor alpha, whilst Th type 2 cells are involved mainly in B cell differentiation and maturation through interleukin 4, interleukin 5, and interleukin 6. Many have established, works have established that there are three different signals for T cell activation. So signal 1 is initiated by the binding of allo-recognition or the process antigen presented by self-MHC, major histocompatibility complex, to the T cell receptor CD3 complex. Signal 2 for T cell activation is provided by interaction between a number of accessory molecule ligand pairs on antigen presenting cells and on APC and on T cells, such as that between CD28 molecule on T lymphocyte with its ligand B7 on surface of antigen presenting cell. On specific ligand binding, a signal delivered that a signal is delivered that acts synergistically with T cell receptor induced signals to produce T cell activation. These signals trigger the CD4 T cell trigger the CD4 positive T cell to activate the cytokine interleukin 2 or IL-2 and IL-2 receptor gene expression, leading to the expression of other cytokines. Then cytokines interplay their receptors, and receptors provide a signal 3, which causes the entire cascade of T cell activation to proceed, leading to a cell division. If a T cell receptor is triggered without an accompanying second signal, then the cell is driven into anergic basis, in which it is not only inactivated but also becomes refractory to the full range of activating signals. Again, there's works by Waldmann that has highlighted sort of the basis in and around and aspects in and around the immunological basis around this. So further, allograft in context of allograft rejection is usually considered to function of signal 1, and the grafted tissue or allografted tissues are rejected because they express foreign antigens. However, a great deal of evidence suggests that the signal 2 is also important. So, for example, ovaries, kidneys, thyroid, and pancreas can still be successfully transplanted or allografted if they first carefully purged of passenger antigen presenting cells, or APCs, and if fresh donor type APCs are induced at the time of transplantation or soon post, the allografts are rapidly rejected, showing that their failure to act as immunogens is not due to the lack of expressed antigen. More essentially, the antigen presenting cells deplete grafts gradually the APC depleted grafts or allografts gradually induce tolerance to themselves, such that the recipient eventually is unable to reject allografts even when immunized with fresh AP antigen presenting cells. This tolerance is not a generalized suppression, but because there is still an inability to respond to donor type spleen cells, showing that tolerance induced by thyroid or pancreas, for example, does not extend to cover other tissues, and including the spleen. So further to this is that while there are new generation of immunosuppressive agents, is largely on merit of research performed by in context of pharmacotherapy and pharmacology. The understanding of basic mechanisms of the immune recognition and regulatory networks is still a first basis in context of underpinning research. T cells can recognize and react against an allograft or graft, either directly, where this is referred to direct recognition pathway, or indirectly, where there's an indirect recognition pathway. And it has been shown that both pathways mediate the growth and differentiation of allo-antibody producing B cells and contribute to acute and chronic rejection. However, both pathways can be inhibited by particular subsets of specialized T cells, which display regulatory and suppressor function. Regulatory T cells regulatory T cells display a characteristic CD4 positive, CD4 positive and CD25 positive phenotype, and act as natural inhibitors of T cell reactivity to autoantigens. Natural T cells, or also being referred to as natural T cells that are developed or arise in the thymus, are present at birth and persist through adult life. Upon triggering of T cell receptors, they interact with other T cells, inhibiting a response to any kind of stimulation. The inhibition is not antigen specific, and thus it is unlikely expansion of naturally occurring CD4 positive or CD25 positive T cells may find a clinical application in strategies aimed to induce antigen specific tolerance. Antigen specific tolerance in and around where T cell activation, allorecognition, and response and allo-response in organ transplantation is concerned. And so therefore in and around this, regulatory regulatory T cells are derived from the CD8 subset of lymphocytes, and have also been described, have also been documented in the literature. They resemble T receptor type 1 cells, producing interleukin 10 and acting in an antigen non-specific manner. Although in some systems, in some aspects, on some basis of the of and in around immunobiology, in around immunobiology, although in some systems they appear to be antigen specific. Again, antigen specificity is of significance for contemplating in the use of CD8 positive T receptor cells or T cells in and around organ transplantation. In contrast to an immune response to a pathogen in which all of the foreign derived peptides are expressed with MHC molecules, as allograft expresses its own set of MHC molecules capable of priming recipient T cells through the direct allorecognition direct allorecognition pathway. Self-restricted T cell clones can indeed recognize allergenic MHC peptide complexes, the crystal structure of a T cell receptor bound to its allergenic MHC peptide. In literature, sort of inform and confirm this, and show that the interaction is essentially identical to the interaction of a T cell receptor or TCR with a foreign peptide bound to a self-MHC molecule. Early post-transplant or early post-organ transplantation, in the inflammatory environment created by the operative by trauma, in context of trauma, recipient CD4 positive T cells may become activated through direct recognition of alloantigens expressed on the membrane of passenger donor antigen presenting cells. TH cells may proliferate, produce and induce a cascade of cytokines, and elicit the production of CD8 cytotoxic T lymphocytes, which would damage the allograft, causing an acute rejection or acute allograft rejection episode. A successful immunosuppressive therapy will and can reverse rejection, inhibiting the proliferation of alloreactive CD4 positive T helper cells. Donor antigen presenting cells migrates out of the allograft, which thus becomes less immunogenic. Host dendritic cells or DC cells, host dendritic cells or DCs ingest apoptic and necrotic cells of the allograft and process them into peptides and present them to T cells from regional lymph nodes. At the same time, however, under the coverage of immunosuppression, CD4 positive T cells that recognize donor allopeptides may be unable to secrete interleukin 2 and proliferate. In contrast, CD8 positive and CD8 negative T cells, which do not require co-stimulation through B7 or CD440 molecules, may still receive the activation in context of signal and signal 1. Interacting with non-professional antigen presenting cells or APC of the graft of the allograft, i.e. with endothelial cells or ECs, or with autologous dendritic cells, which have have processed donor alloantigens. The prevalence of this population may prevent the cytotoxic activity. CD8 positive, CD8 28 positive cytotoxic lymphocytes, or CTLs, and since endothelial cells do not, or ECs do not, express the co-stimulatory B7 molecules. CD8 positive, CD8 negative, CD28 negative T cells, T cells may also, in line to T cells, may also inhibit the indirect pathway tolerizing the host antigen presenting cell, which present allopeptides bound to MHC class 2 antigens to CD4 positive T helper cells, hence CD8 positive and CD28 negative T cells. T cells may maintain the early quiescence predominantly. So although immunologic tolerance has been induced successfully in laboratory rodent models, by numerous protocols and investigations, including treatment with anti-CD4, anti-CD25, and anti-cytotoxic lymphocyte 4A or CTL4A, and anti-CD40L antibodies and anti-CD40L antibodies. No equivalent success has been obtained. Has been obtained markedly in humans, and certain experimental procedures, investigations, however, have yielded encouraging findings, yet have been abandoned for various reasons. Examples include the protocol. There's works by Strober, which was based on the use of total lymphoid irradiation, followed by the use of low-dose immunosuppression. Although complete and stable tolerance has been recognized to be inducible by this procedure, the risk involved were more than the advantages, or the potential advantages. Similarly, the use of donor specific transfusions in around transplant of donor transplantation in context of donor bone marrow cells have yielded promising findings, yet stable tolerance cannot always be achieved or identified. And so transfusion bone marrow cells may induce a generation of regulatory T cells and or microchimerism, yet none of the phenomena appear to bypass the requirement of iatrogenically induced immunosuppression, or IST. And there's works by Cortesini in 2003 that sort of delve into some aspects in around this. Presentation of self-peptides bound to MHC molecule plays a significant possibility in the process of negative and positive selection of immature T cells in the developing thymus. Similarly, in adult T cell appreciation of self-peptides continuously displayed on antigen presenting cells regulate the immune response to antigens and intrigue the balance of self-tolerance in the periphery. Works by Olliwall in around 2003 have delved into this. Self-reactive T cells that have somehow escaped thymic negative selection or recognize antigens expressed only extra-thymically are further subjected to control in the periphery by either T cell energy, T cell ignorance, and or indifference or deletion upon encounter with self-antigens and the periphery. However, anergic or ignorant or again sort of ignorant T cell populations have the potential to be activated while self antigens are released into circulation during the course of an infection, or when they are activated by cross reactive epitopes present on infectious agents. Evidence has also increased in the past sort of decade or more sort of retrospectively and sort of works in context of literature sort of more prospectively suggests there's a dominant control machinery or mechanisms in which a distinct T cell subset actively downregulates the activation and proliferation of self-reactive T cells that have escaped passive mechanisms of tolerance. In rodent or laboratory models, strains that do not produce autoimmunity, a variety of non-specific autoimmune diseases can be initiated by interfering with normal T cell growth, or by rendering the animals moderately T cell deficient. Further, since adopted transfer of a well-defined T cell subset from syngenic healthy donors can prevent the development of autoimmunity in lymphopenic recipients, it is reasonable to suggest that normal immune system contains immunoregulatory T cells that can prevent the activation of auto-reactive T cells. Thus, findings in works by Olliwall and team indicated in around their works that thymic selection may not delete self-reactive, but also give rise to regulatory T cells, presumably specific for self-antigens in the case of natural self-tolerance, or regulatory T cells specific for allo-antigens in the case of organ transplantation and transplant tolerance. Some of self-antigens are effectively invisible to the immune system, immune system in context of immunological, and where there's immunological ignorance. This can occur and occurs because the antigen is sequestered in an avascular organ, such as the vitreous humor of the eye. Immunological ignorance also occurs because CD4 positive T cells will only recognize antigens presented in association with MHC class 2 molecules on professional antigen presenting cells, or APCs, and means that most organ specific molecules will not be presented at levels high enough to induce T cell activation. Those works by Underwood in 2000 in and around this. Self-antigens and lymphocytes are also kept separate by restricted routes of lymphocyte circulation, which limit naive lymphocytes to secondary lymphoid tissue and the blood. To preclude large to preclude or prevent large quantities of self-antigen from gaining access to antigen presenting cells, debris from self-tissue breakdown must be cleared rapidly and destroyed, and this is achieved by cell death through apoptosis, so preventing widespread spilling of cell contents, together with a variety of scavenger mechanisms which look to mop up, so to speak, cell debris. So, in aligned to this, all of the sort of problems described in around in around sort of the works that have been highlighted directly or indirectly to the fact that current immunosuppression, in context of current immunosuppression agents, and again in around T cell activation, allo-recognition, allo-response, and organ transplantation, the immunosuppressive agents tend to be more non-selective in their in context of pharmacotherapy action. Chronic rejection could probably be prevented. Chronic rejection could probably be prevented by administration of very high doses of immunosuppression, but this would inevitably lead to unacceptable increases in transplant associated infection and or other comorbidities, other complications. The full potential transplantation of transplantation will only be realized when alternatives to non-selective immunosuppression are identified or found. The major aim of transplantation or transplant immunobiology or immunology is to develop procedures or protocols or investigations that prevent immune responses towards the allograft but leave the rest of the immune system intact. Transplantation tolerance appears to offer the best hope of achieving this effectiveness and specificity. Studies in intrathymic models have provided important information on the mechanisms of acquired transplantation tolerance, but intrathymic intrathymic injection, for example, might not be an ideal route to tolerance in context of in context of clinical transplantations for two main reasons. So first, because the first being that the majority of T cell differentiation takes place during fetal development and in very early neonatal period, and it is not clear, is not transparent or clear whether the introduction of allogenic tissue into adult human thymus would shape the T cell repertoire in the same way as it does in laboratory rodent models. Second, both lobes of the thymus must be exposed to guarantee efficient injection of alloantigen. This involves opening the chest through a procedure, sternotomy. And although intrathymic injection of alloantigen is certainly possible during routine cardiac transplantation or cardiac organ transplantation, and has been conducted or carried out in at least two patients, in at least two patients, it's unlikely that the risk of such a procedure would be or could be considered worthwhile for other major organs. Most of the clonal deletion of T cells in the thymus is driven by bone marrow derived cells, probably dendritic cells, present self-antigens of developing T cells. If male mouse of the B10 strain is crossed with a female of the genetically distinct genetically distinct strain, a distinct strain referred to as CBA strain, bone marrow cells of the developing B10 aligned to the CBA strain in the fetus migrate to the thymus and present B10 and CBA antigens to maturing T cells. The resulting F1, the resulting the result in and around this is where murine or mouse models have highlighted therefore tolerant. There is some aspects of tolerance that have been identified of both B10 and CBA in context of CBA strain and in and around self antigens. So the question is then, a question then also arises. Would it be possible to interplay the powerful mechanisms of central tolerance if donor, in context of donor and bone marrow, could be introduced into the peripheral circulation? And so, in line to this, for many years it has been possible to generate bone marrow chimeras in rodent laboratory models, and indeed such models provided some of the earliest evidence for positive and negative selection of maturing T cells. Similarly, approaches later have applied to organ transplantation and organ transplantation models in which bone marrow depleted recipient strain murine or mice were reconstituted with a mixture of both donor and recipient bone marrow. These mixed allogenic chimeras, which are genetically unaltered by bone marrow infusion, except donor skin grafts or allografts with immunosuppression. Donor skin grafts without immunosuppression, but are capable of normal B cell and T cell responses to other challenges. Although they therefore exhibit true transplantation tolerance, most of these models, laboratory investigation laboratory models, required whole body irradiation to allow the injected bone marrow to become established or identified, and such an approach is probably unacceptable in routine transplantation or routine clinical transplantation. However, recent works, more recent works has identified that it is possible to achieve a basis of mixed chimerism and transplant tolerance with a much less aggressive conditioning regimen in both murine and primate transplant models. These procedures and protocols of investigations involve transient inhibition of T cell function and low dose irradiation, followed by intravenous infusion with donor bone marrow. Success in experimental models, laboratory models, have led to several attempts in clinical transplantation to augment conventional immunosuppressive protocols and procedures with conventional immunosuppressive pharmacotherapy with peripheral administration of donor bone marrow. Some of the results are encouraging, but overall the rates of rejection have not have not been reduced significantly. Several reasons could account for this, including one, the inability of the bone marrow cells to migrate efficiently to the thymus, two, the deficient maturation and thymic export of T cells, in the adult, and or three, the disruption of delicate tolerance mechanisms by the co-administration of immunosuppressive pharmacotherapy. So now, in and around some contexts around interleukin 2 and interleukin 15 in context of organ, and in context of organ transplantation. So in acute allograft rejection within the nephrology discipline or specialty, there have been intrigues in and at the same time to acknowledge the most essential interleukins in transplantation. For example, interleukin 2 and interleukin 15, which have recently, which have been cited for have been cited or linked have been linked for in context of significance. So interleukin 2, also known as T cell growth factor or TGF, is secreted by stimulated T helper cells, CD4 positive, cytotoxic T cells, CD8 positive, and granular lymphocytes, large granular lymphocytes or LGL. And it promotes interleukin 2 promotes proliferation, or where there's a colonal expansion and differentiation of additional CD4 positive cells, B cells, and activates macrophages and oligodendrocytes. Works by Waldmann have highlighted some aspects in around interleukin 2 and interleukin 15 functions. The latter distinguished in the initial stimulation of the proliferation of activated T and B cells on antigenic stimulation, as well as the upholding and commencement of natural killer cells or NK cells. However, the two cytokines also provide complementary assistance to the adaptive immune responses mediated by lymphocytes. Interleukin 2 is pivotally implicated in the orientation of self-reactive T cells and thereby facilitate the initiation of tolerance to self. In contrast, interleukin 15 inhibits this interleukin 2 induced process. Furthermore, promote the maintenance of CD8 positive memory phenotype T cells, whereas interleukin 2 inhibits and prevents perseverance in vivo. A crucial challenge that still remains and to be resolved is to define how these cytokines that share two signaling pathway receptor subunits and utilize a common signaling pathway can manifest contrasting functions. In addition, Waldmann and colleagues have highlighted that interleukin 15 signaling primarily occurs by special processes in which monocytes and dendritic cells form contacts with T cells. Forms contact with T cells. Within this immunological synapse, the interleukin 15 signal occurs in association with other signals to the T cells originating from binding of CD40 to its ligand CD40L, T cell receptor TCR to the major histocompatibility complex MHC peptide, and CD28 to CD86. These investigations on the mechanism of action of interleukin-15 have focused on the particular role played by interleukin-15 receptor alpha in the actions of interleukin-15. Interleukin-15 has provided a meaningful immunological investigations. Interleukin-15 has provided meaningful therapy for neoplasia in preclinical models, and research has been carried out to induce it into clinical trials in therapeutics aligned pharmacotherapeutics, in which parenterally injected interleukin-2 has been approved as a basis of pharmacotherapy. The uncontrolled expression of interleukin-15 carries with it a risk to the organism of the survival of autoreactive T cells that could lead to development of autoimmune diseases. Interleukin-15 is potentially a dangerous cytokine in that it inhibits the self-tolerance mediated and facilitates the survival of CD8 positive memory T cells. Survival of CD8 positive memory T cell, including self-directed memory cells. Furthermore, interleukin-15 initiates the inflammatory factors tumor necrosis factor alpha and interleukin-1 beta. In regards to regulation of cytokine expression, interleukin-2 is produced by activated T cells, and its synthesis is controlled at the levels of messenger RNA transcription and stabilization. In contrast, there is a complex of multifaceted regulation of interleukin-15 expression in monocytes and dendritic cells. Interleukin-15 is controlled predominantly post-transcriptionally at levels of translation and intracellular cytokine trafficking. This works by Waldmann again, and the team have highlighted that a blockade of interleukin-15 action have resulted in a decrease in the number of virus-specific CD8 positive cells, supporting the view that in humans, as in murine or mice models, interleukin-15 is crucial for the maintenance of effector memory CD8 positive lymphocytes, and by disparity, addition of antibodies to interleukin-2 or its private interleukin-2 receptor alpha did not or has not had the same same effect or this effect. A series of biological agents that block the action of inflammatory molecules, particularly a tubular necrosis factor alpha, are being utilized or have been utilized in immunotherapy of particular autoimmune diseases. In other immunomodulating approach, most of the previous pharmacotherapy or pharmacotherapy or pharmacotherapeutics directed at cytokine receptors are targeted at the interleukin-2 receptor alpha chain. This approach is based on the examination or investigation that interleukin-2 receptor alpha is not expressed by normal quiescent cells, but it is but is articulated by abnormal T cells that are implicated in conditions, particular leukemias, and select autoimmune disarray and allograft rejection incidents. For future for in context of in context of sort of prospectively, Waldmann and team have highlighted that targeting interleukin 15 or IL-15, IL-15 is a base is involved is a base is involved in the inflammatory cascade, acting as a major stimulus for tuberculosis factor alpha synthesis, and because it has been shown to be implemented in various autoimmune disorders. The disruption of interleukin 15 action might interrupt both the inflammatory components and the self-directed immunological memory that categorize autoimmune disorders. The autoimmune sort of bases that will be that are bases of the initial focus of in around interleukin 15 directed therapy are those where elevated serum levels of interleukin 15 have been confirmed, and in some cases where inhibition of interleukin 15 or IL-15 action progresses the disease in animal models. In particular, major efforts have been encouraged to initiate the evaluation of the cytokine IL-15 as a therapeutic agent for recipients with renal cell tumors and metastatic malignant melanoma, as well as an adjuvant component of cancer-based adjuvant component of cancer-based regimens, as well. Now, to highlight a little bit around co-stimulation and in around co-stimulation and around this, there is again works by Matt Zinger in the early 90s have highlighted some aspects around co-stimulation in their works, tolerance, danger, and the extended family in context of immunology or immunobiology, and also that there has been more works around co-stimulation in literature more prospectively. While recipient T cells recognize MHC, major histocompatibility complex peptide complexes or complexes through the direct and indirect pathways, this recognition event is not sufficient to induce full activation of the T cells. Full activation requires antigen presentation by professional antigen presenting cells, for example, or i.e. a dendritic cell capable of delivering a stimulus, capable of delivering a simultaneous second signal of the T cell, and this is referred to as co-stimulation. And so co-stimulation can be mediated through a number of molecules. Several have recently been been identified, but two molecules in particular appear to have a pivotal basis or a pivotal role. CD28, where the ligand on T cell interacts with B7-1 or B7 and or B7-2, also identified as CD80 or CD86, accordingly on the antigen presenting cell, and CD40 ligand or CD40L, also referred to and known as CD154, accordingly on the T cell interacts on the T cell, which interacts with a CD40 and interacts with CD40 on the antigen presenting cells. So if a T cell binds to an MHC peptide complex in the absence of co-stimulation, this can potentially prevent the T cell from becoming activated, alter the T cell function so that it develops into a regulatory or suppressor cell, or induce apoptosis of the T cell. And this phenomenon is also referred to as co-stimulatory blockade, as works by Heger in early 2000 that delve into this as well. Developing pharmacotherapy regimens such as CT cytotoxic lymphocytes, lymphocyte A4 or CTLA4 Ig, a chimeric molecule consisting of common two leukocyte antigen 4 CTLA4, a concentrated molecule antigen of T leukocyte and a portion of the human immunoglobulin on the human immunoglobulin common region Ig, which blocks the CD28B7 interaction, and anti-CD40L antibody, which blocks CD40 and CD40L interaction, are progressively efficient at preventing and precluding T cell activation, and may also induce true immunological basis in around true immunological tolerance. These molecules are still being investigated currently and sort of prospectively in around various various investigations, various works aligned to immuno in around transplantation and transplant immunobiology, in order to identify and establish effects on prolonging human allograft survival. So in line to this, also just to iterate a little bit around NKG2D. Human NKG2D was originally identified in the early 90s as an orphan receptor that is expressed on natural killer cells and many T cells. Natural killer cells express a large number of different cell surface receptors that deliver either activating or inhibitory signals. The relative balance of these signals regulates NK cell activity. Many of these receptors also occur on gamma theta T cells and on activated alpha beta T cells. NKG2D is also an activating receptor that has recently generated intrigue. A number of NKG2D target ligands have been identified, and the most intriguing of these have been paired with closely related proteins called or referred to MICA and MICB, which interplay with major histocompatibility complex MHC class 1 chains, or class where there's an interlink with MHC class 1, where there's an interplay or interlink with MHC class 1, and these are cell surface molecules distantly related to MHC class 1 proteins, and the genes possess elements of heat shock promoters. MICA and MICB therefore are expressed during cell stress and upregulated in tumor cells and during viral infections. This receptor ligand combination may play a critical role in the immune response to a variety of pathologies. The NKG2D genes exist within NK complex on human chromosome 12 and mouse or murine chromosome 6. Genes within this complex encode structurally similarly type 2 lectin-like receptors, and many of these are expressed primarily in NK cells. Experiments or investigations with soluble NKG2D proteins demonstrate that the mouse NKG2D ligands are expressed at appreciable levels on thymocytes, but at low levels or not at all on most normal tissues. Expression is upregulated, however, on many tumor cells. Expression has been observed or identified in renal allografts undergoing acute allograft rejection. Acute allograft rejection. So that now draws this audio podcast to a close. This has been an audio podcast aligned to the Ztorm Focus v.3 and the T cell activation, allo-recognition, and allo-response in around organ transplantation or transplantation, and also where this audio podcast is aligned to observing chemokine and chemokine receptor expression in acute allograft rejection, or AAR. This has been another Ztorm Robotics production. So thank you for tuning in.